Hi, I'm Alaina Smith, and I'm going to tell you how insulin is produced. Insulin is produced by the pancreas, if you didn't know that, but we're not actually going to produce insulin. Instead of using insulin, we're going to use the GFP gene, the green fluorescent protein, to show throughout the lab where insulin is shown when it's out with the UV light. Okay, well, the first thing you're going to do is you're gonna have your 4 petri dishes, and you're gonna have your 4 tubes. Your 4 tubes are gonna have the P glow. There's gonna be 2 of them that are negative and 2 that are positive. The 2 that are negative, one's gonna have P glow LB, and the second one's gonna have P glow LB in amp. The positive ones are gonna have P glow LB in amp, and the second one is gonna have P glow LB amp and arabinose. That's the part that's gonna switch over to the green fluorescent light, the GFP, to trigger to glow. So after you have that, what you're gonna do is you're gonna add the transformation, the CaCl2, which is calcium chloride, and you're gonna add the P glow plasmid to each of the tubes. After you've done that, what you're going to do is you're going to put the little tubes on little things like this, and you're gonna put it in the heat shock, and then afterwards you're gonna freeze it so that it closes all of it together so nothing goes anywhere. After you do that, after you've done that, what you're gonna do is you're gonna get an inoculating loop, and you're gonna get the stuff that you got the day before, and you're going to take it out and you're going to spread it on the petri dish. I'm not going to take it out and show you, but I'm going to represent as much as I can. You're going to take it out, you're going to open the petri dish, and you're going to spread it across. And make sure you have the correct label one so you don't cross-contaminate or mess up any of the order. Afterwards, what you're going to do is, after you're done spreading with all different inoculating loops, you're going to tape all these together to make sure nothing gets out or nothing opens or it doesn't get knocked over by other classmates. You're going to tape it all together, put it in the incubator for about a day, and when you come back the next day for class, it should be finished. And just make sure your teacher set up all this. If not, just ask. And afterwards, when you come next day for the class, you should have your petri dishes ready to start part 2. Okay, so now that we've taken these, the petri dishes out of the incubator after leaving them for a day, what you're going to do is you're going to get the UV light, okay? Then you're going to see which of the colonies that are in there that glow. Now, the ones that's going to glow, I can already tell you, is going to be the LB, the AMP, and the arabinose, because the arabinose is what triggers the GFP to glow. So after you realize that, you're going to get 2 tubes. You're going to label both of them LB, AMP, and arabinose. One's going to be the negative and one's going to be the positive. What you're going to do is you're going to get another inoculating loop, and you're going to get the one that glows, the colony that grows, and you'll get a colony and you'll put it in the positive, and you'll get another inoculating loop to do it again with the same one to put it in the negative. And after you've done both of those, what you're going to do is you're going to get tin foil. You're going to wrap these to where no light can get inside of these. And what you'll do is, after you've done wrap them, you'll put them back in the incubator. Well, they never were before, but you're going to put them in the incubator horizontally, just like this, so that the bacteria that's inside of it can have a complete surface area to grow, so that nothing is contained or anything, to make sure everything's grown. And you're going to leave that there for a day, and then when you come back to class the next day, you'll get them out and start the next part of the lab. Okay, so after you take these out of the incubator, you're going to unwrap the foil that was around them, and what you're going to do is you're going to get a micropipetter. You're going to get the correct one that fits, and then you're going to take whatever's in here out, and you're going to put them into the correct labeled clear tubes that you've labeled yourself. After you've done that, what you're going to do is you're going to centrifuge these for about 90 seconds so that the pellet and the supernatant separate so you can see what is what. And after you do that, you're gonna dump the supernatant out of both of the tubes, and you're gonna add TE buffer. What the TE buffer is gonna do, you're gonna mix it a little bit, and then you're gonna shine the UV light to see what glows in the tube. Afterwards, you're gonna add the lysosome protein, which all you're gonna do is you're gonna freeze it overnight so that the bacteria ruptures inside of here. And when you come back the next day, back into class, you're going to centrifuge this so that the protein stays on top, and whatever's left in there that's not protein will go straight to the bottom, so you can see the difference that you have in your tubes. Okay, so our next step is to get the supernatant out of this, right? And you're gonna pour it into this. And what you want to do is, when you're gonna end up cutting this, you're gonna want to make sure that this is open so that it's gonna let it drip, because otherwise if it's closed it keeps the air shut inside. And what that's gonna pour into, you're gonna have 3 tubes that you've labeled 1, 2, and 3, and you're gonna put each of your columns into one of them. You can either hold it, a partner can hold it, or you guys can like hold it with like a slided piece of paper right there so it stays there, and you're gonna have it drip there for about 5 minutes. Well, what you're gonna do is, after you've already put in the supernatant to the 3 columns, you're gonna add the binding buffer. And the binding buffer you're going to want to put on the sides. You're going to want to try to put it on the sides as much as possible so that it will get rid of all the high hydrophilic stuff of the waste all the way to the bottom so that it comes out. Because what you want to do is you want to get all the stuff that's not needed in the bottom or whatever. And the next thing you're going to add is you're going to add the wash buffer. The wash buffer is also hydrophilic, and it's just going to get rid of the extra stuff that didn't go down with the binding buffer. And now the last thing you're going to add is the elution buffer, which the elution buffer is very hydrophobic, and it's going to get rid of all the hydrophobic stuff inside of here. Because what you're going to do is you're going to put it on the sides again once again, and that's going to help isolate the protein, and that's pretty much it. Alright, after you've already emptied all of the stuff from the protein chromatography that came into these 3 tubes from earlier, what you're going to do is you're going to take the stuff that's out of here, and put them into these tubes. And after you've done that, after you've labeled the correct HIC tubes, there's going to be 3 of them. 3 HIC tubes. You're going to add 20 microliters of the samples that you've had and the sample of the loading buffers. After you've done that, what you're going to do is you're going to put these in one of these little things again, and you're going to go heat it in a little container that's 95 degrees Celsius. You're going to heat it for about 5 minutes. After you've done that, what you're going to do is you're going to set this up. Now, this is for the protein electrophoresis. This is going to give you your final end result of the lab. What you're going to do is you're going to take the lid off, and you're going to fill it up with water all the way to the top, to about where it says like 4 gels. When you see it, you'll see what it's talking about. And you're going to get, and the stuff that for the gels, you're going to get a little package. It comes in a little package deal, and you're going to hook it up in there in the container, and make sure you take the green part on the bottom, otherwise your thing's not going to work. And make sure your wells are on the right side. Now, after you've done this, you've already set this up for about 5 minutes. This is already going to be done. You're going to come take this out, and you're going to cool it. And after you cool it for a couple minutes, you're going to take the stuff out of here, and you're going to what you're going to do is you're going to get one of those, like we had earlier, one of these. But it's going to be very skinny and very small, so it fits inside the gels. You're going to take the stuff out of here, and the samples that your teacher gives you, that's what you're also going to take. And you're going to have the gels from 1 through 7. Now, 1 and 7 you don't fill up with anything, because those are the ones that are already like done, and those are going to be the original stuff that you don't mess with. 2 through 6 is what you're going to end up filling. You're going to end up filling it with the 3 HIC tubes that you have, and the extra sample that your teacher gives you. After you've done this, your teacher's going to, after you set this whole thing up, you're going to tell your teacher, and your teacher's going to set this up to the machine that it's supposed to go to, and it's going to run for about 35 minutes. And after it's done, it's going to give you your end result, and you're going to see the protein electrophoresis ending. I hope you enjoyed the lab.